Okay, so I've labeled this part B of the week 4 lab, and it's kind of odd because these PowerPoint slides show up earlier in order on the general PowerPoint, but I wanted to give an idea of the structure of the organization of the skeletal system before we start looking at this microscopic structure of bone, because we're going to be looking at long bones. So in the previous video, we talked about how the long bone had a hardened middle shaft called a diaphysis and expanded ends called epiphysis. And so this right here, this bone's been cut, and you can kind of see, we talked about the articular cartilage on this. This is the bottom end, the distal end of a femur. And so what we've done here is we've cut the diaphysis in half, so you can see the inside of it. We talked about it a little bit on the slide or the video previously. So I've got this hollow middle cavity called the medullary cavity. Endosteum lining the spongy bone on the inside. There'd be yellow bone marrow in there, blah, blah, blah. But what we've done with this, as you can kind of see, is they've taken a pie wedge slice of this and expanded it into this. So out in here would be the medullary cavity. All right. So we're going to look at how this little pie wedge part is put together and see the intricate workings of the compact bone. But I'm going to enlarge that little pie wedge so we can see it a lot easier. So on this, one of the things to see first and foremost is there's a dividing line kind of right here. All of this on this side is what we call compact bone. On this side is spongy bone. Now on the spongy bone, there's not a lot we're going to point out, but I just want to kind of show that the trabeculae it's pointing at here. I know sometimes it's hard to envision it. It's just, again, in spongy bone, we have these little webs of bone, and they called it bone plates, but the name for those are trabeculae. And so that's where that comes from. So now most of this is going to be talking about the compact bone. Now, to start with, all compact bone has a hard, very tough connective tissue membrane around the outside called the periosteum. Now, on this picture, they don't see it. You'll see it in a model picture that I'll show you in just a minute. But there are little anchors, little things. They almost look like it almost looks like a drawing of static electricity. They're called Sharpie's fibers, and they are little fibers that are attached, the periosteum, to the compact bone. And again, you don't see them on this picture, but we will when we see the model. Now, the real defining feature of the compact bone are these repeating circular structures called osteons. This is what gives bone its really impressive what we call tensile strength, which means the ability to resist breaking. So if I was to go out and take a tree limb, a real thin tree limb off of a tree that's alive and tried to break it, it wouldn't snap. Now, if I found one that was on the ground and has been off the tree for a long time, I could bend it and it would pop pretty easily. The tree, the fresh tree limb has what we call tensile strength that can resist bending at a point of breaking, and bone has a really impressive tensile strength, and it's all because of this repeating osteon that we're going to look at. Now, one of the things is we'll need to look at is even the osteons. They're made up of layers. You can see that these are multiple layers. That's why people say that bone kind of looks like tree rings, right? Trees that are cut acrossways. These little rings, these are called lamella. Again, I'm hoping that you have your student handout sheets with you there. These, you'll see that they're called lamella. Now, lamella is another way of the Greeks' language of layers. We talked about strata and different things like that. Lamella are another way of layers, and so this is layered. And the lamella form these little circles to form together to work to make an osteon. Now, on the model, you're going to see that there's a different type of lamella, and you can kind of see them back here. that are around the outside of the bone in order to give it its shape. And on our model, these are the ones that are raised up. You'll see it in a second. I'll point it out. They look like three little billboards. Because it's hard to try and label these individual little layers that are in here, if I labeled it, if I had a picture, I'd probably be pointing to the lamella on there. And so those big billboard lamella on there. So just wanted to point that out. So we've got all of these layers, these lamella, and in between those layers we find these little cells called osteocytes. Now on this picture it looks like a whole bunch of little ants. Most students agree with me when that's where I got it. Most students will call these, they look like little bugs all over this. They are the actual bone cells and they're called osteocytes. Now here's the thing about them, and it's kind of hard to see sometimes. The osteocytes are going to have an outer, how do I want to say this? They are going to be between the lamella. So if you can imagine, like if the wall in your house is a lamella, and you stand up against the wall and someone puts another wall in front of you and you're kind of caught between those two walls, you would be the equivalent of an osteocyte. And so if I draw over here, if I have, I'll put two lamella here. And then I have on this area, I'm going to put this little osteocyte. Boonk, boonk. Right. So the lamella will have to make a little indentation to fit the osteocyte. That indentation itself is called a lacuna. It's kind of like a cocoon. So if I was to go in here and erase my nice little osteocyte, what would be left is this little hollow opening in here. That hollow opening is a lacuna. So it's hard to describe, and sometimes it's hard to visualize the fact that they're two different things. The cell itself is the osteocyte, so we'll pretend like what I just drew here, this is an osteocyte. But the cavity that it is in is the lamella. All right. Now I'm going to show you on the model. On our models, we'll have one section of the model that will have the dark, really black osteocytes filled in, and then other sections of the model, the majority of the model, the osteocyte has been taken out, and you can see that it's a little hollow indentation, and that would be the lamella, or the lacuna. I hope I said that right. So the lacuna, and it's again, you can kind of think of it's a cocoon. It's kind of surrounding it. All right. Now we're going to talk in just a minute about these little legs, but I just want to make sure we're good with the lamella and the lacuna. So as these osteocytes are living in between these walls, you definitely need blood to get to them. They are alive, and all things that are alive need oxygen. And so we have this intricate system of canals to get oxygen to these cells. Now it starts with, for us right now, what I want to start with is this thing right here that's called a central canal. That's what it's listed here as, a central canal. Every osteon has a canal in the center of it. Now I would encourage you not to learn it as the central canal, but the osteonic canal. That's the more scientific name. Now we're going to look at things in the nervous system and the spinal cord. You'll see there's another thing called a central canal. The central canal is a very ambiguous name. Osteonic canal is very specific. So if you know that it's the osteonic canal, you know that it's the canal in the osteon. Just saying it's a canal in the center doesn't really mean much. A lot of people, when they learn it just as the central canal and they start to go label on the practical, they end up labeling the medullary cavity as the central canal. So I caution you, it doesn't matter, central canal is correct if you put it, but I caution you to be leery of learning it that way. Again, the osteonic canal is a good way to learn it because it is very specific. But whatever the case may be, you can see these right here are shown where they're cut in half. So you can see the blood vessels going back and forth in the osteonic / central canal. Now, they are going to be connected to the blood source, these guys right here that are in the medullary cavity, and the blood is going to be working its way out through little parallel holes or canals called perforating canals. So the perforating canals throughout the diaphysis in the bone, you're going to have these perforating canals cutting across. They join with the osteonic / central canals and work on bringing the blood throughout the compact bone. Oddly enough, bone is one of the best blood supplied tissues in the body. And so this perforating canal, as it cuts across and it joins with these osteonic or central canals, they will form kind of a capillary bed, and blood will start to seep out. Plasma, that is, will start to seep out, and that's what's carrying the oxygen, all that yummy oxygen trying to get out to those cells. And this plasma, this fluid carrying the oxygen, is going to seep through the lamella through little pores called caniculi. Those are those legs that you see here. All right, so I'm going to explain what's going on. Now, I don't know how many people who are listening to this have ever lived up north. Up north, it's much more common to have a basement in the house. But if you've ever grown up with a basement, whether you're up north or here, a lot of times the basement is what we refer to as masonry block. If you're taking the classes on the Grand Strand campus, those big blocks of stone that make up the walls there. Well, the thing about those blocks seem very solid. They're very heavy, very sturdy, but they have a lot of little pores in them. They are not good at keeping out water, and that was one of the things that if you live up north, if you knew anything about it, a lot of times it's your parents dealing with it, but if you know anything about it, you'll know that they have to constantly waterproof that, because no matter how strong and how solid they seem, they are very porous. And that's exactly how the lamella are. So the fluid seeps through all of these walls, these lamella, by way of caniculi. Now the caniculi are going to have a much more prominent appearance next to the osteocytes, because the osteocytes are going to be pulling that fluid in. And so as they keep pulling fluid in, those canals that are constantly being used are going to widen, so they become more apparent. So these little ant-like legs that you see all through here are little canals called caniculi that are allowing the blood, the oxygen, the fluid carrying the oxygen to get to them. And then they turn around and go back out the same way. It's a pretty amazing setup as it all works. So we've got these osteons with these osteonic or central canals in the middle, and they're surrounded by these rings called lamella. In between the lamella are osteocytes. Those osteocytes are drinking in fluid and getting, oxygen in, getting the carbon dioxide out. And that is happening through these little pores called caniculi. The caniculi allow the fluid to seep through the lamella. It's going to get back into that osteonic / central canal, join into the blood vessels there, and those blood vessels will work their way out through the perforating canals until they move out of the bone. So hopefully that makes sense on the basic buildup of the compact bone. Now I'm going to go through brief. So this is our model. On this one side, you can see 3 different versions of the osteocytes is the way they do it. This part here, the osteocytes are filled in with dark color, so you see them. They're white here, and I don't know why that is. I don't know, but it's still filled, so they're osteocytes. Now, I don't know if you can tell, but all of these are little hollow openings. The osteocyte has been removed, so those are going to be the lacuna, the little hollow openings that the osteocytes live in. Now over here you can see what I was talking about with those Sharpie's fibers, those white little fibers that are holding that periosteum onto the outside of the compact bone. And then up here you can see one of the the raised up lamella that's on the outside. You can see one behind it here. I'm going to move to the next. Oh, well, while it's on this, you can see the perforating canal. These little guys here are the perforating canals. The ones that are going up and down, those are going to be the blood vessels that are in the osteonic canals or the central canals. And these two guys here that you see, these are going to be osteons. They're not little wedding cakes. They kind of look like it, but they are the actual osteons. So let's see what the next picture looks like. Oh, this is going to just show it a little bit more close in that section, showing the osteocytes, the periosteum with the Sharpie's fibers. From above, now you can kind of see my little wedding cake buddies. Even have little candles up top, but those are actually supposed to be little blood vessels. They're divided up so you can see different ways that they're made. We're not worried about the different things, but this one in particular over here on the right shows how each lamella, in order to make it even stronger, each lamella's fibers run at 90-degree angles from each other. And again, that increases the strength of what those osteons help provide. And again, you can see the osteocytes here. The rest of this model, pretty much everything outside of here, all of these, they're pretty much hollow, and you can kind of see it. And so they're the lacuna. From the overview, you can see this little section. This would kind of be the outer part. Now, in real life, the osteons aren't as nice and organized as it looks here. But that's the osteon. You see the central or the osteonic canal.